In this video, we will study about ion exchange chromatography. Okay, the ion exchange chromatography is a technique used for the separation of ions. That is, charged particles can be separated. Okay, the charged particles. The particles can be separated using the ion exchange chromatography, and also the polar molecules can be separated using the ion exchange chromatography, and also the bulkier molecules like proteins, amino acid and nucleotides can be separated using the ion exchange chromatography. But you cannot separate the hydrophobic particle. Okay, the hydrophobic molecules cannot be separated. Okay, they cannot be separated using the ion exchange chromatography. So, the ion exchange chromatography is mainly used for the separation of what? Charged particles, polar molecules, and proteins, amino acids, and nucleotides. But hydrophobic particles cannot be separated using the ion exchange chromatography. Well, now we will see how the setup used in the ion exchange chromatography. Okay. The setup used in the ion exchange chromatography is like this. Okay, you have the chamber like this. Okay, it is a chamber used in the ion exchange chromatography, and inside this you will be having the stationary phase. The stationary phase in the ion exchange chromatography look like beads. Okay, it is made up of polymeric material. Okay, it is made up what? Complex polymeric material. Okay, we will see about this in the later classes, how you can manufacture such resins. Okay, so so in this chamber what we are having the stationary phase. The stationary phase is like what? Like a bead. Okay, let me draw it here. Okay, it is in the form of bead, and it is having the fixed charge. Okay, the fixed charge is there, and this is connected to the exchangeable ion. Okay, the exchangeable ion, I am representing it by the letter X. Okay, what is X? X is nothing but a exchangeable ion. Okay, the exchangeable ions. Okay, X is the exchangeable ion, while this is what? This is having fixed charge. Okay, it is a polymeric matrix having the fixed charge, and this bond is there no between this matrix and this exchangeable ion. This is ionic in nature. Okay, so ionic bond. Okay, now what can be this X? The X can be cation or it can be anion. Okay, so exchangeable ion can be either cation or anion. Whenever exchangeable ion is cation, we call it as cation exchange resin. Okay, or cation exchanger. Okay, whenever you have exchangeable ion as anion, then we call it as a anion exchange resin or anion exchanger. Okay, we will see how we can synthesize the cation exchange resin and anion exchange resin in the later classes. Okay, so now you understand that this X exchangeable ion can be cation, like what? Like it can be H plus or it can be sodium plus, like that. Okay, anion can be chloride or OH minus, like that. Okay, so whenever you have anion, we call it as anion exchanger, and whenever you have cation as exchangeable ion, we call it as cation exchange resin. Okay, now we will understand the principle of the cation exchange resin. Okay, that means we are understanding the principle of the ion exchange chromatography by taking the example of cation exchanger. Okay, now consider you have the mixture of three ions. Say you have the mixture of lithium plus, and you have the mixture of sodium plus and K plus. Okay, you have the mixture of these three cations. Okay, now what is your task? You need to separate it. Okay, it is initially present together, and your task is to separate them. Okay, That means you have to separate the lithium in one beaker, sodium ion in another beaker, potassium in the another beaker, like that. You need to separate it so that you will be, going for the ion exchange chromatography. Here you need to use the cation exchange resin, because you need to separate what? Cations, right? So you need to go for cation exchange resin. Okay, now I'll draw it here. Okay, this is the chamber, and inside this chamber you have the cation exchanger, okay, or resin, cation exchange resin. Now, how will it be the bead? I told it is in the form of sphere or spherical bead, and the exchangeable ion is what? Cations now. Okay, in case of cation exchange resin, usually you will be having the hydrogen as the exchangeable ion, and also you can have sodium also. Okay, so this is what? This is polymeric matrix. Well, now what you need to do is you first prepare the solution of these ion. Okay, the first prepare what? The solution of these ion in the suitable solvent. Okay, you prepare the solution of these ion in the suitable solvent. Then what you do, you have to pour this solution to the chamber that is containing cation exchanger. So now what happens is that these ions are bigger in size when compared to the hydrogen. Correct? The hydrogen is small in size, atomic number is 1, while these are having higher atomic number, that means higher size is there. That's why what happens is that these ions will have the higher preference for the matrix than the hydrogen. Okay, what I told? So there are mainly two factors. Okay, there are mainly two factors that will decide the affinity of the ions to the resin or matrix. That is, one is the size. When the size of the ion is bigger, it has a greater affinity for the resin, and next is the charge. Okay, the higher the charge, it has a greater affinity for the resin. So what are the two factors that will affect the affinity? One is the size. Okay, the higher the size, it has got higher affinity, and the higher the charge, it has got higher affinity for the resin. Okay, now what happens is that when you pour the mixture of the lithium ion, sodium ion and potassium ion to this chamber, this H, as I already said, the bond is ionic. Okay, it will break, and in this place of hydrogen, hydrogen get exchanged by these ions. Okay, I will draw it here. Okay, this is the matrix. And now in the place of H, what we have? We have the ions of our solution. Okay, here it may be a lithium ion, here sodium, here it may be a potassium, here may be some other lithium, and here it may be some sodium, like that. Okay, there are many resin beads here, so that all hydrogen will be replaced by what? Lithium or sodium or potassium. Okay, now in the chamber I have this thing, lithium, sodium and potassium. Now how can I separate them from the resin? So what I have to do is I have to wash this chamber using the solvent, using the solvent of little higher concentration. Higher concentration solvent has to be added to this chamber, so that what happens, this ions that is attached to the matrix can be separated. Okay, say I'm washing it with the say one normal HCl. So when I put the one normal HCl, maybe I can replace the lithium that is attached to the matrix, because the lithium, when you compare to the rest of the ions that is present in the solution, the lithium ion is smaller than the sodium ion, and which is smaller than potassium ion. So when you have lithium here, and when you add the higher concentration of say HCl, okay, one normal HCl, okay, just for example I'm telling. Okay, when you pour the one normal HCl to the chamber, now this is having higher concentration. So what happened? This H plus can replace the lithium that is attached to here. So now in the place of lithium, what I have, I'll be having the hydrogen of the solvent that I have used. Okay, let me erase this. This is now in the place of lithium. What I have the uh, let me write it. Here in the place of lithium, now I have hydrogen. Okay, the lithium is replaced by the hydrogen. Now, where does that lithium go? It will go uh, and it will be eluted out. Okay, it will be eluted out first. Okay, when you compare to these three ion, the lithium is having smaller size. So, lithium is the one that is getting eluted out first. Okay, now still we have sodium and potassium that is attached to the resin. Now what I have to do, the one normal HCl is not enough for that. I have to increase the concentration of HCl. Say I'm increasing it to say 2 normal HCl. Okay, what I'm doing, I'm just pouring the 2 normal HCl to this chamber. Okay, now this hydrogen is having higher concentration, so it have the greater affinity for the resin. So the sodium can be replaced by the 2 normal HCl. Okay, I will erase that sodium. Wait a minute. So sodium will be now erased, or in that place what I have, I'll be having the hydrogen. Hydrogen from where? Hydrogen from the 2 normal HCl. So now where does that sodium go? The sodium will be removed from the matrix and it will be taken out, or it will be eluted out. Okay, sodium is eluted out. Now still we have the potassium. Okay, so to remove the bigger sized ion, we need to have the higher sized or a higher concentration of acid. Say I'm increasing to 3 normal HCl. Okay, just for example purpose, I'm taking this concentration. Okay, so I'm just increasing the concentration of HCl and I'm pouring it to the chamber. Now the potassium can also be replaced by the hydrogen of the solvent. Okay, wait a minute. I will erase that potassium, and in that place of potassium, now what I have, I will be having the hydrogen. Hydrogen from where? Hydrogen from the 3 normal HCl. So the potassium will be cleaved or removed from the matrix and it will be collected or it will be eluted out. Okay, and it is taken in a separate beaker. Now see, all ions which initially it was together, now it is present in different beakers. So this is how you can separate the ions using the ion exchange chromatography. Okay, so this is about the principle. Okay, if you have any doubt in this topic, please let me know in the comment section. Okay, in the next class we will study about how we can prepare the ion exchange resin, like cation exchange resin and anion exchange resin. Okay, thank you for watching.